Good morning, everyone. This is Dr. Manasa DJ, Assistant Professor, Department of Studies in Botany, Davangere University, Davangere. I'm very happy to be a part of this international virtual conference on biodiversity and ecosystem services in a climate change perspective. I'll be presenting my work entitled as Extrication of the Hypotriploid human cell line suppressive and photocatalytic degradation activities of biofabricated copper oxide nanostructures which were synthesized by Tabernamentana hainiana phytochemicals. Introduction. As we know that these days textile industries have generated a lot of effluents which includes carcinogenic dyes, bleaches, oil, grease, sulfides, phenols and chromiums, etc. All these effluents which are going to compromise the quality of water bodies by increasing the biological oxygen demand, chemical oxygen demand, which is impairing the photosynthesis, inhibit plant growth, etc., thereby entering the food chain, and they are responsible for the bioaccumulation of these heavy metals and other things which will promote toxicity, mutagenicity, and carcinogenicity. So, our aim was to degrade these carcinogenic dyes which are eluted by the textile industries, which are hazardous to the living beings. So, in our work, we have studied the photocatalytic degradation of these catalytic dyes, carcinogenic dyes, using copper oxide nanoparticles, as well as these days lung cancer is also a leading cause of global death, so it's a major healthcare problem in India. About 13% of all new cancer cases reported are of lung cancer cell lines. Hence, we have also tested the anti-cancer property of the synthesized nanoparticles using the nanoparticles synthesized by the plant extract. Objectives of the present study are to synthesize and characterize copper oxide nanoparticles using leaf extracts of Tabernamentana hainiana wall, also to evaluate the photocatalytic and anti-cancer activity of copper oxide nanoparticles. Coming to the plant which I have taken for my study, this is the plant Tabernamentana hainiana wall, which belongs to the family Apocynaceae. The plant is a small deciduous tree which bears the milky white latex. The flowers are white in color and it bears the follicles, fruits called follicles. Methodology. First step was the collection of plants from the Western Ghats. Next, the extract was prepared from the collected plant samples by thorough washing in running tap water. The washed plant samples was dried at room temperature and it was powdered, and the extract was obtained by refluxing the plant extract using water at 80 degrees Celsius for about 2 days. Then after the extract was obtained, the extract was tested for the presence of polyphenols as well as the ferric reducing antioxidant power assay, that is antioxidant assay. Antioxidant activity was determined. Then the copper oxide nanoparticles were synthesized using solution combustion synthesis method. Synthesized nanoparticles were characterized using various different characterization tools. Once the copper oxide nanoparticles were characterized, the nanoparticles were tested for their photocatalytic dye degradation activity as well as anti-cancer activity. Coming to the results of my experiments, in this picture you can see the antioxidant activity of the plant extract. Here ascorbic acid was used as a standard. In the graph you can see with the increase in the concentration of plant extract, the antioxidant activity was also found to be increasing. Correspondingly, the absorption of the plant extract which was being tested for antioxidant activity was also being increased. the color gets intensified with the increase in the plant extract concentration. So the total phenolic acid content was found to be 43.18 to 79.09 milligram per gram gallic acid equivalent weight. The IC50 value of the antioxidant activity was found to be 183 microgram per ml. Coming to the characterization results of FTIR, the image A shows the spectra of the plant extract, whereas B shows the spectra of the copper oxide nanoparticles synthesized. Here you can see various absorption peaks in the spectra as well as in the nanoparticles spectra also. You can observe the presence of polyphenolic compounds, aliphatic compounds, C double bond C stretching groups, vibrations which are responsible for flavone derivatives. You can also see the spectra for like bands for carboxylic acids, phenolic compounds, etc. All these bands seems to have to be shifted slightly because they have involved in the capping and reduction of the copper oxide metallic ions. In the nanoparticle spectra, you can observe transmission peaks at around 540, 545 and 530 centimeter inverse, which is responsible for the copper oxide nanoparticle stretching. Coming to the results of powder X-ray diffraction studies, in this X-ray XRD spectra, various absorption peaks have been observed. All the obtained absorption peaks corresponds to the monoclinical structure of the nanoparticles obtained. In the B picture, you can see with the increase in the concentration of extract, the peak has gone sharpening and narrowing at the base and sharpening at the tip. This is because of the influence of plant extract used for the synthesis purpose. Coming to the SEM images, results and elemental composition studies. Looking at the images, you can observe that these are the lower magnification images and these are higher magnification images. In the higher magnification images, you can observe the highly agglomerated non-homogeneous type of nanostructures obtained. This is because of the solution constant synthesis method effect. These pores are obtained because of the liberation of gases during the process. And all the samples obtained are found to be spherical in shape. The elemental composition studies showed that the presence of absorption peaks which are corresponding to copper as well as oxygen molecules. Here you can also observe the spectrum absorption peak for carbon, which is because of the plant extract used for the synthesis purpose. The results of UV visible spectroscopic analysis. Here, when you take a UV visible spectra, you can observe few bands which are responsible for the presence of polyphenols present in the plant extract, whereas this image, B image, shows the surface plasmon resonance absorption peaks of the copper oxide nanoparticles synthesized after the process. These bands which are obtained at 373, 391, 378 are responsible for the surface plasmon resonance bands of the copper oxide nanoparticles. Energy band gap was calculated using top plot and the band gap energy was found to be 3.47, 3.6, 3.96 electron volt correspondingly. Coming to the dye degradation studies, in these images you can see this images shows the percentage dye degradation versus irradiation time. Here, with the increase in the irradiation time, the dye concentration in the mixture goes on decreasing. That means the nanoparticles have degraded the dyes which are present in the mixture. These first two images are of methyl blue dye degradation studies results, and these two are the dye degradation studies for methyl orange. You can observe around 90 to 92 to 97% dye was degraded within a span of 180 minutes in both the cases. So, all these results favor that the copper oxide nanoparticles exhibit very good efficiencies of dye degradation studies because of their smaller size as well as optimum band gap values. To the anti-cancer study of prepared copper oxide nanoparticles, these are the phase contrast images of the anti-cancer studies. Anti-cancer studies was carried out by employing MTT assay. All these images, cisplatin was used as a positive control. You can observe here with the increase in the concentration of plant copper oxide nanoparticles, the cell viability percentage was decreasing. All these images shows the variegated cell morphology such as cell bursting, clumping of cell, loss of cell membrane integrity and organelle damages within the cell. Conclusion goes like this. Here we have developed an efficient, effective, rapid and cost-effective method for the synthesis of nanoparticles employing solution combustion synthesis method. We have also shown about 90 to 93% dye degradation efficiencies of the nanoparticles against the carcinogenic dye. Also, we have checked the recyclability profile of the nanoparticles and the results show even after the fifth cycle of experiment, the nanoparticles can be reused for further experiments. Effective catalyst dosage was found to be 20 to 30 milligram. Hence, this method could be used in the treatment of industrial effluents on a large scale. Also, nanoparticles exhibited significant anti-cancer activity which could be used against the treatment of